All right, it is my great pleasure to introduce our PI speaker for today, Dr. Sevinc Ercan. Sevinc is originally from Turkey, where she did her undergraduate degree. She then moved to the US for graduate school at Pennsylvania State University, where she studied transcription using yeast as a model system. After this, she began her work on understanding organization of the genome during her postdoctoral work at the University of North Carolina, and she has continued this work in her own lab at New York University. Her lab is interested in understanding the molecular mechanisms that specify and regulate large chromosomal domains and their effects on transcription. And she uses two model systems, primarily nematodes as well as yeast. So without further ado, I'll hand it over to Sevinc to hear what I think is our first nematode talk at Fragile Nucleosome.
Thank you so much, Pravruta, and rest of the Fragile Nucleosome organizers for giving me a chance to present our work here. And today I'm going to talk about Condensin DC. All right, so one of the many fascinating aspects of development is that during these cell divisions, the chromosomes undergo drastic changes in structure to support both chromosome segregation and precise regulation of transcription. And in animals, one of the key mechanisms by which chromosomes are compacted during cell division is through making DNA loops. And the protein complexes that make these DNA loops belong to the evolutionary conserved protein complex family called structural maintenance of chromosomes complexes. These are SMC complexes. And the chromosome compaction in mitosis is regulated by a SMC complex called condensin. So the current hypothesis by which condensins form loops is through this loop extrusion hypothesis, in which the condensin holds the DNA and makes progressively larger loops using the energy from its ATPase activity. All right, so what's the evidence for loop extrusion? There are different lines of evidence. One is a very compelling video of this loop extrusion happening in vitro on DNA. The other being mathematical models simulating the data that one can achieve from high-C experiments in vivo. So some of the features of the high-C data in vivo can be predicted using the loop extrusion simulations in computer. All right, so far, pretty good. But what we don't understand yet is that, well, actually, how does condensin translocate on DNA in vivo, and what regulates the condensin translocation on chromatin, and what's the function of these DNA loops in gene regulation? So we want to address these questions using our favorite model organism, Celecans. Like other animals, Celecans have two canonical condensins, condensins 1 and 2, and these complexes bind to all chromosomes during mitosis and meiosis in both sexes to regulate chromosome condensation. In Celecans, there's a third condensin. Okay, so this condensin differs from condensin 1 by a single subunit. So the remaining 4 subunits are the same. So this subunit called DAMP27 has duplicated and diverged and acquired a different function. So condensin DC, what it does is it binds to X chromosomes across cell cycle, only in hermaphrodites, to regulate dosage compensation. And that's why I'm referring to it as condensin DC. So let me introduce the system a little bit, okay? So how does the condensin DC only bind to the X chromosomes? So the way that we think this happens is that X chromosomes contain a number of sequence-specific recruitment elements, and these elements are in part defined by a short DNA sequence motif. And the recruitment elements, there are strong ones and there are weak ones, And the strong ones are X-specific, and they typically contain multiple copies of this motif. We don't know what binds to this motif, but a reasonable assumption is that these proteins that I'm showing you here that are required for the condensin to bind to the X chromosome, so these are what we call recruiters, might be binding to these sequences. All right, so once the complex is recruited, it actually coats the entire chromosome, okay? And so what it does is there are about 3,000 genes on the 17 megabase X chromosome, and it binds to many of the expressed genes on the X. And so we know that this spreading is linear. And the way we know that is through a really powerful tool that is the X autosome fusions that we can use in Celecans. So in these fusions, the complex binds to the X chromosome and then spreads into the autosome, and the spreading reduces with distance from the nearest recruitment sites, okay? So the spreading is linear and reduces from distance from the recruitment elements. Okay, so why do you need this nice even distribution of the complex? It's because the function of the complex is to dosage compensate. So what the complex does is it represses transcription initiation from the X chromosome promoters by about twofold in hermaphrodites, so that you have overall X chromosome transcription from 2X animals equal to the single X males. All right, so hopefully you're thinking to yourself, okay, I'm convinced that this is a good-looking condensin with reasonable knowledge about how it binds to the chromosome. Now we can address the in vivo translocation and its regulation. So the first question we asked is condensin DC spreading consistent with the loop extrusion hypothesis? To address this question, David and June, two graduate students in the lab, decided to perform Hi-C. So some of the features of the Hi-C maps in C. elegans X chromosomes are supported by the loop extrusion hypothesis, and that's the presence of topologically associating domains, these loop kind of structures forming, and these flames and stripes, etc. And so one thing that I want to point out here is the topologically associated domain boundaries in C. elegans correspond to some of the strong recruitment sites, which loop and interact with each other. All right, so how does the interactions on the X chromosome look? And are they different from autosomes? Yes, they are. And we can see this in a distance decay curve plotting the interaction distances across all the autosomes, 1 through 5 on the X chromosome, and X kind of stands out here, right? So we wanted to look at what are the specific 3D DNA interactions. What you can do is you can take the decay curve for the X chromosomal bins and divide it up with the autosomes. And what you can see here is you can highlight the X-enriched 3D DNA contacts, and on the X chromosome, these contacts are between 100 kb to a megabase length. All right. Several papers from other labs also defined some of the properties of the Hi-C data on the C. elegans X chromosome. The first being from the Meyer lab, that show that the condensin DC recruiter SDC2 knockdown abolishes the TADs on the X chromosomes, and the strong Rex sites, about 8 of them, form some of the TAD boundaries on the X. And Chankowski, Corsis, and Raleigh labs show that the condensin DC binds to loop anchor sites on the X chromosome, and we've also recently shown that actually condensin DC binding and the Rex sites are sufficient to form loop anchored TADs if inserted onto an autosome. So far, the data is reasonably consistent with a loop extrusion model with notable differences, which we can discuss later if you would like. So next, the question is what regulates the processivity, the translocation of condensin DC? So for this, we thought about what might be some of the regulators, and got inspired from some of the work from Hirano lab, right? So what they did is they asked, what are the minimal components that we can use to build a mitotic looking like structure in vitro, okay? So the two major components that are not histones and chaperones are condensin 1 and topoisomerase II. So, and at the same time, there were several studies that actually suggested that perhaps the topoisomerases might be important for regulating loop extrusion. So the idea is this. The hypothesis is that loop extrusion would locally concentrate these DNA supercoils or catenets, and these will serve as reaction centers for topoisomerase II. And topoisomerase II would relieve these catenets so that loop extrusion can proceed unhindered. So our approach was very simple. We wanted to remove TOP2 and see what happens. And this project is led by a postdoc in the lab, Ana Morao, with the help of Jun, who has done the high C for this. Okay, so what Ana did is first develop the Auxin inducible degradation system for Celecans top2. So, and she checked, okay, how long does it take for the top2 protein to get degraded? And in 1 hour, the amount goes down a lot. Okay, so what is this remaining? It is the protein in the germline. So dosage compensation complex, the condensin DC, binds and regulates transcription in somatic cells. So we didn't want to deal with any of the germline issues, okay? So we are specifically degrading TOP2 only in somatic cells. And she made sure that this is the case. And she saw that after 1 hour that of Auxin treatment, there is no more TOP2 in the somatic nuclei. All right, so the prediction of the hypothesis is that if TOP2 relieves the supercoils and catenates, then the loop extrusion can proceed unhindered, okay? So if that's the case, then the prediction is the condensin DC processivity should decrease in the absence of TOP2. Is that the case? When we look at the high C data, it appears that that might be the case. The first thing that you will notice are these flames coming off of the REX sites are getting shortened. And you can also see that the X-enriched 3D DNA contacts get shorter. So here's in gray and black are the control data, where you can see that the X-enriched 3D DNA loops, DNA contacts are in the, when you get rid of TOP2 is getting shorter. But what was most striking to me is this next data. That is that if we look at condensin DC binding across the X chromosome, we looked at this and we're like, oh my God, this looks like mountains, okay? Mountains around the strong REX sites. So what we see is that in the control data, where we're looking at DAMP27 binding, ChIP-seq, so condensin DC binding along the X chromosome, what you can see is that relatively even binding with these strong recruitment sites being bound a lot more. But if you get rid of TOP2, you kind of see this localization, accumulation around the strong REX sites, recruitment sites. Okay, so the question was that, how does TOP2 promote loop extrusion? Is it supercoiling or is it the DNA catenets? So to address this question, what Ana decided to do is look at another topoisomerase. And this one is going to be topoisomerase I, and in Celecans, that's TOP1. So the TOP1, unlike TOP2, cannot remove DNA catenets. It can only take care of supercoils. So the idea is that if the DNA catenets are preventing the processivity of the complex, then in a top-1 knockdown, we should not see that appearing of the mountains. All right, so what happens? So Anna did the Auxin inducible degradation for top-1. In 1 hour, again, top-1 was gone in the somatic cells. And when she did the DAMP27 ChIP-seq and compared to the top-2, she did not see that same kind of accumulation around the strong Rex sites. So the conclusion is that the DNA catenates are the ones that reduce long-range translocation of condensin DC. All right. But what Anna noticed is that in a top-1 minus, something different is happening. And you can see this when you zoom in the data, okay? So when you zoom in the data, in the wild type, what you can see here is, again, condensin DC binding. And we know that the binding is mostly enriched at the promoters of actively transcribed genes. In a top-1 depletion, you start seeing this accumulation, okay? So what's going on here? The complex is being accumulated at the gene bodies. So what might be going on here, we think, might be something to do with transcription. So transcription is known to induce supercoiling, right? Ahead of the polymerase, you have positive supercoiling, and behind the polymerase, you have negative supercoiling. So maybe top-1 is dealing with these supercoils. And in the absence of top-1, condensin DC is kind of getting stuck. All right. If that's the case, we would actually expect genes that are expressed and longer to have a little bit more problem and maybe show more top-1 accumulation. And that's kind of what we see. We see that in the absence of top-1, condensin DC is accumulating at the gene bodies and more on the long genes. If you get rid of top-2, you don't see this kind of same gene body accumulation. But if you get rid of both top-1 and top-2, you see a more drastic problem with condensin DC movement. And we interpret this as top-1 and top-2 both dealing with supercoils in the genes, with top-1 being the major regulator. All right. So the conclusion from this is that transcription-induced supercoiling reduce short-range translocation of condensin DC. Okay, so let me go back to that first part of the hypothesis, which was that loop extrusion locally concentrates supercoils and catenates, and these serve as the reaction center for top-2. And the prediction from this hypothesis is that top-2 should localize to the recruitment sites because these recruitment sites serve as tag boundaries by blocking condensin DC passage. Is that the case? What Anna found is that, yes, it's indeed the case. So this is one of the recruitment sites. And what you can see is that top-1, top-2, and DAMP27 condensin DC binds to this site. If we insert a recruitment site, a strong recruitment site on the X chromosome, what we can see is that this recruitment site now is bound by top-2. All right. But if we delete a recruitment site, what we can see is that top-2 binding at this deleted site is now eliminated. So from this, what we can conclude is that the recruitment elements on the X, these recruitment sites, recruit both condensin DC and top-2. All right, so I'm going to now present our current model. All of this data is not published yet, so I welcome any comments and input. So our model is this. What we're thinking is that condensin DC spreads along the X chromosome, and the long-range spreading is actually consistent with some kind of loop extrusion. And but there might be also a short-range spreading that's within gene scale. All right, in a top-2 depletion, what we think is happening is that DNA catenates are preventing the complex to spread out, translocate, but that short-range spreading is not actually affected. In the top 1 deletion, this long-range spreading is not affected, but in fact, there is a problem with the short-range movement of the condensin DC within the genes. So with that, I'd like to thank my lab, who are amazing. Anna did all the top experiments. June did the high C. C was a master student who helped Anna characterize the degradation strains for top 2 and top 1. And I'd like to thank our collaborators, especially Paul Maddox, who gave us the top 2 antibody in Celecans. Anna is on the job market, and if you want to hear a little bit more about the Topoisomerases, she's actually giving a talk at the New York City Chromatin Club that you can watch on October 19th. And with that, I can stop and take any questions.
Thank you, Sevinc. That was wonderful. We have a few questions in the Q&A. I'll remind the audience again that you can raise your hand if you have the ability to talk, and we can unmute you. And also, if you want to chat with either or both speakers, please let us know in the chat, and you can stay afterwards for coffee chat with the speakers. All right, I'll start with the questions in the Q&A. Jan Sorsinski, I'm so sorry, asks, does condensin DC have a WAPL-like release factor?
We actually don't know if condensin DC has a release factor. So that's basically the answer. It would be lovely to find out if there is.
And then we have a question. This is from an anonymous attendee who asks, do you see loss of corner spots or loops in the high in the absence of top?
Let me see if I can actually. Yeah. Yeah, the answer is yes, we do see this. So I want to show you this data here, hopefully that you can see it. So when we look at the Rex to Rex loops, in the wild type, you can see this, and in the mutant, it's gone. We don't know how to interpret this just yet. But that's what happens.
Yeah. Okay. Matthew Levine says, hi, Sevinc, very interesting model there. Are you saying that transcription impairment in top 1 AID cells is at the origin of condensin redistribution?
I am not sure if I understand the question very well. It is it at the origin of condensin redistribution? If you can clarify, maybe I will have a better idea how to answer it.
Okay, so, I think Matthew is also staying for coffee. So if you want to raise your hand, please do, or you can talk more about this in the coffee chat. Peter Meister asks, what are the phenotypes for the top 1-2 degron strains? Do you see any DC related phenotypes?
Yeah, we actually don't look at these worms after an hour because if you knock down top 1 or top 2 by RNAi, they're dead. That's the phenotype. Okay. So we can't really say much about that. After 1 hour knockdown, when we do RNA-seq, we do see X upregulation. That means that both top 1 and top 2 are contributing to X chromosome repression. Yeah.
Mm-hmm. Okay. Nikolai Zolotarev asks, how conserved is this function of top 2 in mammals?
I expect everything to be conserved 100% in mammals. But it's very reasonable to think that they are conserved because there are decades of research that have actually linked top 2 and condensin in regulating mitotic chromosome separation. So if you get rid of top 2 or if you get rid of condensin, what you get is those DNA catenets, and they make these DNA bridges that have trouble segregating. And so you can think of, you can think whatever we're seeing here is actually mechanically conserved in regular old mitotic chromosome. But of course, we would love to go and see if that's the case.
Okay, and I have a kind of related evolution question, I guess. You said the dumpy 27 was a duplication. Is that something that is true across nematodes?
It is true for CNR rhabditis. We actually can detect dumpy 27 by sequence in CNR rhabditis, but outside it, we actually don't, we are not able to detect it by blast. So one thing that one of the graduate students in my lab is really interested in is figuring out in another nematode like Pacificus, which is plant parasite, is this thing conserved? So he's doing some experiment to address that.
Okay, cool. We have one more question that's come in the chat. It's from Chaitra Sanjeevamurthy, who says, very interesting talk. In which phase of the cell cycle did you check the condensin and topoisomerases binding?
Yeah, I didn't clarify this, but this was all done in early larval tree stage of Celecans. There's not much going on. The cell divisions are done. So these cells are mostly an interface.
Okay. And I think Ben, who's one of the organizers, also has a question. So I'm going to let him unmute himself and ask.
Hey, great talk. Quick questions, and apologize if I missed this, but I was wondering for your Rex, like the Rex binding site insertions, we could then see top 2 there or not. Do you know that's specific for top 2? Like, or do you also see top 1 being recruited there as well?
Yeah, I don't think Anna did the chips, or I haven't seen it yet, but for top 2, it's there. I don't know if top 1 is there, but in regular old wild type, top 1 and top 2 both localize to the recruitment sites. So it would be reasonable to assume that top 1 is going to be recruited as well.
And do you have any sense yet if this is like a physical interaction or something about the topology of the DNA that's leading to the recruitment?
Yeah, so in terms of physical interaction, I think Anna did the experiments, and if I remember correctly, there was not a physical direct interaction. In terms of topological effect, there might be something, because my lab just recently published some supercoiling measurements, you know, these methods like Sorlin cross-linking kind of method, that showed that the recruitment elements had a negative supercoiling in the wild type. And in the absence of condensin DC recruiter, SDC2, that negative supercoiling was gone. So it's possible that there's some topological structures happening at the recruitment sites.
Thank you.
Mm-hmm.
Can I ask a related question? So are top 1 and top 2 also recruited when you briefly mentioned that you put these sites on autosomes and you see recruitment? Is that true for both of them?
Yeah, we didn't look at that yet. It would be nice to be able to see that. I mean, we can make really nice, I mean, not the nicest TAD on an autosome, but it's a pretty good looking TAD. I can show you here. So this is from David's preprint. Basically what we're doing is that we're inserting 3 recruitment elements on chromosome 2. We need 3 because if you put just 1, it's not enough. It doesn't recruit enough condensin. So when we put that, what you can see here is that this is an 80 kb region. So we had to get pretty high resolution IC data. Is like something that looks like a TAD, and you see these loops. So it would be interesting to see if the topoisomerase is going over there as well.
And does it matter where you put these sites on the chromosome?
We didn't have the energy to try too many insertions. Right, but one thing you can tell for sure from another paper we published in 2017 is that you need more than 1. Just having
Okay.
one or two is not enough. And we think that that makes sense in the context of an X-specific binding, because what you don't want to have is you don't want to have a recruitment site on an autosome by mutation or whatever, and then now you recruit lots of complex there, right? So what you do is you build a very cooperative kind of recruitment strategy, so that you can only bind to the X chromosome and not the other chromosomes.
Okay, we do have a raised hand now, or a couple of raised hands. I'm gonna let Jan talk first.
Hi, thanks so much for the beautiful talk. I've just got a quick technical question. So are these experiments done on whole animals, or do you somehow do cell type specific chip?
Oh boy. So, I mean, Celecans is awesome for a lot of things, but for cell type specific, it'll be like, my people will run away from my lab if I propose that. So, yeah, that was a long answer. The short answer is yes, we used whole animals, unfortunately. Yeah. So it's everything is mixed up.
I see, but it's still clear. I mean, it's still homogeneous enough that you see really nice patterns, right?
Yeah, absolutely, right? Like isn't that really surprising? It's surprising and not surprising at the same time. These features are very conserved between cell types. That's what we would see. So that's kind of how I would interpret that.
And I just have a quick second question, again, technical. I mean, it's like, so this compensation, so you see this loading, this is, in adult animals to maintain. And it's kind of, I'm asking if it's similar to how kind of like exist, you have an initiation and the maintenance phase, or do you just kind of always get this loop extrusion throughout the life cycle of the animal?
Yeah, I mean, this is something that we discussed a lot, and we are still discussing. We don't have a very good idea of what some of the earlier events of what's happening in very early embryogenesis. So we don't, but it would not surprise me that there is an establishment and a maintenance phase. And with this kind of degrown systems in larva, we're mostly looking at the maintenance. But I would love to start now going very early embryogenesis and figuring out how do these things work during the establishment.
I see. Thanks so much. It's really fascinating.
Thank you.
Okay, and we have one more raised hand from Colin. You should be able to talk now.
Hi. Yes, fantastic pictures. So I was wondering if the rec sites are orientation dependent. So the orientation effects.
Yeah, one of the remarkable differences between CTCF's type of system and the rec sites is that we don't see a motif orientation dependence. And this was first, we had reported this, we haven't seen it, that kind of motif dependence, but we actually recently experimentally tested that idea by taking a strong recruitment site that has multiple motifs and inserting in two different orientations at the same locus on the X chromosome and asking, is the looping specificity different between these two? And we didn't see anything that's different. So it is independent of the motif orientation.
So do you reckon they can stop loop extrusion in both directions then?
I don't know how the rec sites work as TAD boundaries. They might be stopping loop extrusion in both directions. And how does that happen? We are not sure. We don't know.
Yeah, thanks.
Oh, all right. We are approaching the hour, so I'll just remind people that if you want to join the coffee chat, please let us know in the chat. If not, we'll see you in a couple weeks for the next talk. And also, if you don't let us know in the chat that you want to join, but you're still here, we will eventually throw you out of the coffee chat to try and keep it private. Thank you.